Mechanism of termination of transcription within the prokaryotes can be understood with the help of this slide. As you can see on the slide, we have the double-stranded DNA acting as a template for the transcription. And the mRNA synthesis has already started and it is undergoing the transcription. Almost the process of transcription is complete since the RNA polymerase here it is shown to be approaching the terminator site. When the RNA polymerase is there at a point just near to the termination site, at that point of time, an area that is present on an mRNA molecule is available for attachment of row protein. That area is called as RUT site. RUT site is nothing but row utilization site, that is row factor utilization site. The information for this row utilization site, it is present in the row recognition site present within the DNA. So when this section will undergo the transcription, it will ultimately be represented within the RUT site that is present on the mRNA molecule. As soon as the RNA polymerase is near to the completion of transcription mechanism and it is approaching the terminator site, at that point of time, a row protein, this is a row protein, which is a type of a helicase protein, will come and attach to this RUT site. As soon as this row protein attaches to the RUT site, it will try to move forward in the 3' direction towards the RNA polymerase. When it is moving through this RNA molecule towards the RNA polymerase, the mRNA that is present within this particular section will undergo formation of a secondary structure which is called as stem and a loop shaped structure. Now we know that the stem and loop shaped structures, they are formed only when there is complementarity of certain bases within this section. So due to complementarity of certain nucleotides within this section, the stem and a loop shaped structure or a hairpin shaped structure is formed within this place within an mRNA molecule that is being synthesized. As a result of that, the RNA polymerase enzyme that has nearly completed the transcription process, it will pause. In row-dependent termination, as we have now just discussed, the row protein first attaches to the RUT site, then tries to move forward in the 3' direction. At that point of time, within the newly synthesized mRNA molecule, a stem and a loop-shaped structure is formed. This is due to complementarity of nucleotides within this section of mRNA molecule. Due to formation of the stem and loop-shaped structure, the RNA polymerase, which is now near to the terminator region of a transcription unit, it pauses. Due to pausing of RNA polymerase at this position, the row protein will now try to move forward towards the 3' end. The row protein, as it is moving towards the 3' end, it is responsible for separation of DNA from RNA. Now we know that at this particular point of area, there is the template DNA, the 3' end of newly synthesized mRNA and RNA polymerase together. As soon as the row protein travels from the 5' end to the 3' end of that RNA molecule, all the components of the transcription mechanism, that is the DNA, the section of RNA which is hybridized with the DNA, that is RNA, the row protein and the RNA polymerase, they all get separated from one another, resulting in termination of transcription.